We're gonna go ahead and get started here. First and foremost, thank all of you for braving the cold and coming out this evening. We really appreciate it. Nice crowd here. So tonight we've got a science cafe. Austin Nuxoll is going to talk to us about the undead, in particular undead bacteria and antibiotic resistance. But before we hand the microphone over to him, I just wanted to point out to everybody that we will be starting our membership drive likely next week, and we'd like to encourage anybody who's not a member, who has interest in research, who is currently doing research, to perhaps consider joining our organization. Beyond a line on your CV, it's a good organization to be a part of if you are passionate about scientific research, about sharing with the general public scientific research. That's kind of what we're here for right now. But beyond that, if you're doing research and you've presented research, especially as a student, you might as well get recognized for having done so. And membership in the organization in and of itself is recognition of the work that you've done. Beyond that, Sigma Xi has awards that they offer, so there's actually money out there for the work that you've done potentially, which is a worthwhile endeavor. And beyond that, for just general membership of an organization that is passionate about the same things that you're passionate about. We have a really good group here at UNK, but outside of UNK, there are over 500 different chapters of Sigma Xi, over 60,000 members throughout the entire world. So membership in the organization gets you access to all of that. So be on the lookout for information about our membership drive, and if you're doing research, if you're interested, please talk with myself or one of the officers about how to apply. So back to the topic at hand here, antibiotic resistance. Dr. Nuxoll is a faculty member in biology here at UNK. He actually originally received his bachelor's degree from UNK, went on and earned his PhD at UNMC. Beyond that, he did a postdoc at Northeastern University in Boston, Massachusetts, and then in roughly the span of 8 years, is that what we said? 8 years-ish, then came back here and is now a faculty member in the biology department. So without further ado.
All right, thank you for the introduction and having me give a talk here today. So, like Evan said, my talk is about undead bacteria. And it sounds pretty cool, right? That's probably the coolest part of this talk. Don't get your hopes up too much. But really, most of you have probably heard about antibiotic resistance. And the first part of my talk will focus a little bit on antibiotic resistance, current problems that we're facing with antibiotic resistance, and then future problems. But really, I want to spend most of this talk talking about antibiotic tolerance, which I'm sure most of you are not familiar with. So currently, with antibiotic resistance, we're seeing that it's a major problem. We see it in the news every few weeks and once a month. We see a certain organism that suddenly the news is kind of sensationalizing that we no longer can treat it with antibiotics, which has some truth to it, but in reality, we still have some antibiotics that we can treat these bacteria with, right? Although, there are current studies out there that are trying to forecast how much of a problem this is going to be in the future. So currently in the United States and Europe, we have about 50,000 people die each year from antibiotic-resistant bacteria. If you look worldwide, we have about 700,000. Now, given our current limitations on antibiotic discovery and the rate at which antibiotic resistance is emerging, it's expected by 2050, according to some studies, that there will be 10 million people that die every year from antibiotic-resistant organisms. So a huge jump from the current 700,000 people worldwide. And then I have a map over here showing which continents will be hit the hardest. Asia and Africa both have estimated over 4 million people every year dying from antibiotic-resistant organisms. For comparison, we currently have about 8.2 million people die every year from cancer and 1.5 million people die every year from diabetes. So antibiotic resistance is expected to become a major problem in the future, and a lot of this is because we aren't investing as much as we should in antibiotic discovery, and antibiotic resistance is becoming a major problem. In 2014, President Obama issued an executive order kind of addressing this need. And in 2015, it went into effect where the US government launched the National Action Plan for Combating Antibiotic-Resistant Bacteria. Basically, the main emphasis of this plan was to identify antibiotic-resistant organisms faster in an infection, and then to try and reduce the number of these infections. We didn't always have an appreciation for antibiotic resistance. It really wasn't until the 1990s that we really started to notice. Before this point, doctors often were dismissive of the idea that antibiotic resistance was really a threat. If the first antibiotic didn't work, they had plenty left on hand, they would give them another antibiotic, the infection would clear, they would move on. It wasn't really until they had to move to giving a patient a third antibiotic, a fourth antibiotic, that they would start seeing an effect on the patient. But they really started to shift their attitude and say, maybe this is a problem. Around the same time, towards the late 1980s, multi-drug resistant tuberculosis was becoming a major problem in New York City, and they had to spend over $1 billion trying to control those infections. Media caught wind of this, it started to sensationalize the topic, and suddenly the public really started to pick up on this. And from that, agricultural use of antibiotics kind of came under scrutiny. And currently, we do have protocols that limit certain antibiotics in the United States, but China especially is notorious for giving a lot of antibiotics to their livestock. The purpose for this is to help their chickens grow infection-free, they get bigger, they can make more money off of it, same with cattle and pigs. And actually there was a recent study where they took samples from supermarket meat and looked how often they could find antibiotic-resistant organisms in those samples. Ground turkey, 79% of the time they found antibiotic-resistant organisms. I think it was pork chops had around 70%, ground beef around 60% of the time they had antibiotic-resistant organisms. And then chicken parts like chicken thighs and chicken breast, they were only around 40% of the time that they had antibiotic-resistant organisms. So we're seeing antibiotic-resistant organisms a lot in our meats, right? That's going to be a problem contributing, or a major factor contributing to this problem moving forward. On the right, I have a list of a few antibiotics. Now, this isn't a comprehensive list, but you can see the year that the antibiotic was deployed, and then how long after that it took for resistance to be observed. In some cases, it's only 1 or 2 years after we started using the antibiotic that resistance kind of popped up. Other times, it's maybe a few decades. But the point is, with all of our antibiotics that have been deployed, we've seen resistance, either in limited cases or widespread antibiotic resistance. And these couple of figures here nicely illustrate how antibiotic resistance happens. So on the left, you have a population, maybe it's an infection, maybe it's the bacteria in your gut or your microbiome, but there's going to be a lot of them, several million of bacteria in these environments. Most of those bacteria are going to be drug susceptible, or the antibiotic will work on them. Although you might have a couple, and they're shown in red, that are resistant to the antibiotic. If you start using antibiotics, it's going to kill all of your susceptible bacteria, and you're left with the resistant ones. They're actually going to grow in the presence of the antibiotic, and eventually take over the whole population. And then step 4, you see that antibiotic resistance being passed on to another organism. So a lot of times antibiotic resistance occurs from a mutation in the genome, or they're acquiring DNA from another organism. That's being selected for in the right environment. So you put antibiotics there, you're selecting for that mutation, and eventually they can pass that on to similar species. Now, with the figure on the right, we talked a little bit about this, but you have your agricultural usage of antibiotics. That's going to select for antibiotic-resistant organisms. You either directly consume that meat, especially if it's undercooked, the bacteria are going to survive. They also have to make it through the acidic environment of your stomach, but eventually they can make their way to your intestines, where they can then pass on their genetic material to other similar species. then you have spread of antibiotic resistance. Another way that agricultural use contributes is sometimes you use fertilizer from these animals for crops. Well, you're directly putting feces from these organisms, so they're passing bacteria and that onto crops. If you don't wash your vegetables properly, or it can even spread into water runoff, that's also going to contribute to antibiotic resistance. The other problem is George. We have George here as an example, but we have misusage of antibiotics. So doctors prescribing antibiotics for viral infections, that's a common one. Patients not taking the dosage of antibiotics for as long as they should, or maybe the wrong antibiotics being prescribed, or not enough of the antibiotic. All of that can help select for those antibiotic-resistant organisms. And then if George here visits a healthcare facility like a hospital or a nursing home, uses the bathroom, doesn't wash his hands very well, touches a door handle, and then you have a healthcare worker also touch that door handle, go to other patient rooms, you can suddenly see how this antibiotic resistance spreads pretty easily between the general public. So I've mentioned some of this already, but how did we get to this problem of antibiotic resistance? Well, bacteria by nature have a lot of genetic plasticity. What I mean by that is they easily have mutations that occur. Those mutations can be selected for during antibiotic treatment. They can acquire new DNA easily from other organisms. We have misusage of antibiotics, which I've mentioned some examples of that. My mom was guilty of this, just talking to her a few months ago, and I proceeded to chew her out. She was saying how she had this infection or this cold for a really long time, and she had leftover antibiotics from a year ago. So she just decided to take them to help her get over the infection, right? So the problem with that is, one, she doesn't know if it's a viral or bacterial infection. The antibiotics have been sitting around in a cupboard for over a year, so they're going to have decreased effectiveness. I chewed her out. She kind of was dismissive. That's kind of how our relationship is. But other problems contributing, you have antibiotics from being used with livestock. It's not as much of a problem in the United States, but you can have crowding and poor sanitation. So if you have those antibiotic-resistant organisms in your microbiome and you're using bathrooms, not washing your hands, and you're in a crowded area, you're going to pass those resistant bacteria on to other people that live near you. And the last one is it's kind of a good problem to have, right? We have people who have HIV or organ transplants who have longer life expectancies now, and which is a great thing, but the downside to that is they're often immunosuppressed. They're going to have infections because they're immunosuppressed, therefore they're having more antibiotics used in their lifetime than they would otherwise. So all of these contribute to the problem of antibiotic resistance. So moving forward, it's pretty clear we need more antibiotics, right? So where do we get antibiotics from? Well, there's two broad categories. You can have natural antibiotics, those that are directly coming from the environment, or you can have synthetic antibiotics, those where you're trying to just modify chemical structures, kind of guessing a little bit, trying to figure out what structure might be effective in killing bacteria. With natural products, we often take isolates from soil or marine organisms, and we look for bacteria in those environments that are producing an antibiotic. A lot of times, these bacteria are producing antibiotics to help them outcompete other organisms. We hope to isolate those bacteria, grow them in a lab, take the antibiotics they're making, and hopefully use that against pathogenic bacteria. The problem with this, or one problem with this, is these bacteria that are making the antibiotic are often known as a producer, and in order for them and other bacteria of the same species to survive this antibiotic, they have to have their own intrinsic mechanism of resistance. That means a mechanism of resistance already exists in nature, and it's only a matter of time until that's passed on to other bacteria. Another problem is oftentimes these antibiotics have to be modified to become less toxic to eukaryotic cells. They may have to be more stable. They may not have a long life expectancy. So oftentimes they're modified, making them a semi-synthetic antibiotic. For synthetic antibiotics, it's a little bit of a guessing game, kind of searching for a needle in a haystack, making thousands of different structures, screening those in hopes that one of those structures will kill bacteria and not your eukaryotic cells. So traditionally, when antibiotics were first being discovered, most of those are going to be natural products. We have kind of since run out of identifying natural antibiotics and have moved over to making synthetic antibiotics. The biggest problem with trying to identify natural antibiotics is if we try to take organisms from the environment and grow them, we're really only going to be able to take about 1% of those organisms and actually grow them in a lab. So this means 99% of bacteria and fungal species out there are unable to be utilized for their antibiotics. Well, there was a lab that kind of recognized this limitation, and they also were from Northeastern University, and they developed what was called the isolation chip, or the iChip. And you can see a picture of that here. Each one of these little dots here is an individual well. You can see that blown up here. Each one of these wells has a membrane on the bottom, and then you dilute your sample, whether you're taking a soil sample or a marine sample, you dilute it to where you would expect about one organism per well. You then have a membrane on top, and you put this isolation chamber back into the environment where you took your sample from. So here they're just going to put this right in the soil. What this does is it allows nutrients from that soil and from other organisms in that soil to pass through the membrane. Now the bacteria can grow and produce enough antibiotic that could then target your organism of interest. So if you wanted to find antibiotics against E. coli, you would put E. coli up on this top paper. Or if you wanted antibiotics against Staph aureus, you put staph up there. This method allows us to get that ability to culture organisms closer to about 50%. So now we have a lot more organisms we can culture. There's still a lot of organisms out there that we're not going to be able to do anything with yet. But this invention, about 15 years after this was invented, they had been screening soil samples that whole time, and they eventually found an antibiotic called teixobactam. This antibiotic was, and this was a little bit contested in literature, but the authors claimed the first natural antibiotic discovered in over 30 years. And as a result, it was the most publicized article of 2016. So a lot of press around this article for a couple reasons. One, we're now able to discover new antibiotics. But the other nice thing about this antibiotic is it has two targets in the bacteria. So typically antibiotics will have one target. They may target protein synthesis, replication, cell wall synthesis. The problem with this is bacteria can just have one mutation in that and now they have resistance. The nice thing with teixobactam is it has two targets. So if the bacteria mutate one of those modes of action or sites, there's still another target for teixobactam to bind to where it can kill the organism. So this is great news, right? Now we have antibiotics and we can kill bacteria again. Well, not quite. So, after you discover an antibiotic like teixobactam, it's going to take about 12 years after that for it to get through all of the testing. That's testing how well it can move through the body, is it toxic to eukaryotic cells, you have clinical trials, and then you have regulations from the FDA. So, a long time from now before this actually can do anything. And in reality, it's probably not going to amount to anything because only 1 in 5,000 candidate drugs are ever approved. The other problem is $800 million to over $1 billion is the estimate for what it costs to develop an antibiotic. As a result, we see large pharmaceutical companies now leading antibiotic discovery. We're left with really only small biotech companies and academic labs, and they don't have the resources that these large pharmaceutical companies do. So this is part of the reason in the beginning when I mentioned by 2050 they expect over 10 million people to die every year from antibiotic resistance. And this is just an article, and you look at a lot of sources and you see the same thing. All of big pharmaceutical companies are leaving antibiotic research because it's not profitable. It's much more profitable to invest in cancer, autoimmune diseases, diabetes. So now that I just got done explaining how much of a problem antibiotic resistance is, you're probably thinking, well, we need to invest a lot more money into antibiotic resistance, right? We're looking for new antibiotics. This is only half of the story. It's going to get even more depressing. So you have, if you do a pub search and you use PubMed, which is a major database for looking for biomedical research, and you look for Staph aureus infections, probably 95% of those papers are on methicillin-resistant Staph aureus infections. The vast majority of Staph papers are looking at antibiotic resistance. If you try to get the number of infections for how many infections Staph causes every year, you really only can find stats on methicillin-resistant Staph aureus. You really don't see anything with methicillin-sensitive or susceptible Staph aureus. Here, this is probably 4 or 5 years old from the CDC, but over 80,000 infections from MRSA every year, resulting in over 11,000 deaths every year. Here you can see a picture of a common infection. A lot of times these Staph infections cause what are called minor skin and soft tissue infections, although this doesn't look that minor, but a lot of times it will appear as a boil, maybe a little bit worse than a pimple. So, with everything being emphasized on MRSA, you must think, well, MRSA infections are the vast majority of infections, right? There's limited data, but they looked at 2011, 2012, 2013, 2014, and they looked at what percentage of Staph infections are actually caused by methicillin-resistant Staph strains. And you'll notice it's hovering around that 47%, drops to about 45% in 2014. So really, the majority of Staph infections are caused by methicillin-susceptible Staph aureus, not by resistant organisms. We also see resistant Staph infections dropping over time. So back in 2005, we had over 1,000 infections every year caused by resistant Staph aureus. Going to 2011, we're down to about 80,000 infections every year. So the problem with Staph, methicillin-resistant Staph is actually dropping, and it doesn't make up the majority of infections. So why is there such a discrepancy in the literature? Why are we emphasizing research on methicillin-resistant Staph aureus so much? It must be because it costs a lot more to treat, and there's a higher mortality with it, right? Well, if you look maybe 15 years ago, it was more expensive to treat, and there was a much higher mortality compared to susceptible Staph aureus. However, recent papers actually show that trend to flip. There was a study by Erickson and company, and they were looking at a couple different units in a hospital, and they found of Staph infections, 71% were methicillin susceptible Staph aureus infections. So even more in favor of susceptible Staph than the previous slide showed. Also, if you look at mortality, you really don't see much of a difference. 12% mortality with methicillin-resistant Staph versus 10 with susceptible Staph. If you look at cost, there's really no difference. If you look at susceptible Staph pneumonia cases, about 40,000. If you look at resistant Staph, about 38,000. Overall infections, they're very similar in cost as well. This is kind of confusing, right? We have resistant organisms. In theory, they should be harder to treat. The patients are going to be in the hospital longer. It should be more expensive. They should have a higher mortality associated with them. But we're not seeing that. And it's not really known why this is, but one of the leading thoughts is this is caused by antibiotic tolerance. And you'll hear me say persisters a lot. Persisters are these cells that mediate antibiotic tolerance. They kind of persist in the presence of the antibiotic. And I think this graph nicely illustrates, or this figure nicely illustrates the difference between antibiotic tolerance and antibiotic resistance. So let's start off with resistance first, because that's what most of you are familiar with. Up here, you have a little circle, and in there, it's probably hard to see, but you have individual bacterial cells. All those in gray are going to be sensitive to the antibiotic. Those shown in green are going to be resistant. On the y-axis, you have colony-forming units per mil. This is just a way to enumerate the bacteria. So the higher up on the y-axis, the more bacteria you have. The lower on the y-axis, the fewer bacteria you have. And up here, you probably have about 100 million bacteria. You add an antibiotic, you're going to see most of your population die, right? All of those gray cells in there that are susceptible to the antibiotic are going to die. You have your resistant ones in green, and they actually can start growing in the presence of the antibiotic. So they're going to thrive in the presence of the antibiotics. They're growing, it's like there's nothing there trying to inhibit their growth. Eventually, they're going to pass on that mechanism of antibiotic resistance to their progeny, and you have a whole population that is resistant to that particular antibiotic. If you try to add that same antibiotic again, you're going to see no effect. They're going to flatline. Once the nutrients run out, they survive and thrive in the presence of the antibiotic. Tolerance, on the other hand, that's going to be shown in yellow here, or these persister cells are shown in yellow. You add an antibiotic just like before, you're going to see most of your population dying off, and then you get what's known as this persister plateau, a place where the population just levels off. And these cells are a little bit different. They're not thriving in the presence of the bacteria. They're just hanging out, waiting for an opportunity where they can grow again. And this is kind of where the title comes from, undead bacteria, right? They're not really living. They're just hanging out, waiting for an opportunity to grow again and repopulate. So where you might see this in a clinical setting is during an infection, you're going to have maybe a biofilm-mediated infection. And so for those of you who don't know what a biofilm is, you may encounter this in your daily life. You go to clean your shower drain, and if it's been a while, you reach in and it feels kind of slimy. Maybe in the morning when you wake up and run your tongue along your teeth, it feels a little slimy. That's bacteria growing in those environments, adhering to some kind of surface and to each other by producing a biofilm. Now, where this is a problem in a clinical setting is you have these bacteria adhering to some kind of artificial surface. It could be a catheter, it could be a shunt, it could be a prosthetic device of some kind, maybe a hip replacement, a knee replacement. But you have a small number of bacteria originally introduced, and they start growing and forming this biofilm, and these biofilms are very difficult to treat. But the doctor will notice an infection, give them antibiotics, the infection looks like it's going away, they stop antibiotic treatment. The numbers of bacteria that are living in that environment are so small they can't detect it. A few months later, you have a relapsing infection, and then the infection comes back, so you start antibiotic treatment again, and then it looks like the infection's going away. Maybe a few months later, it comes back. And this relapsing infection occurs in about 20% of these biofilm-mediated infections. Where we think persisters are particularly a problem is when they get down to this persister plateau, it looks like the infection has gone away. There's not a lot of bacteria there, they're not growing, they're not producing virulence factors, so they're not causing a lot of damage to the host. So the doctor will stop the antibiotic treatment. That's shown in blue here, no antibiotics. And now these persister cells can start growing again. Now the difference with these persistent cells or antibiotic tolerance is they're not passing on that mechanism of antibiotic tolerance to their progeny, like we saw with resistance. So once they get to high numbers again, if you were to add that antibiotic again, most of the population is going to die off again. It's always just a small subset of the overall population. Another important difference to know between tolerance and resistance is with resistance, you're going to have a mutation that occurs that allows resistance to a very specific antibiotic. It's often one class of antibiotic. With tolerance, it's a generalized mechanism. So those cells that are tolerant to vancomycin, for example, a type of antibiotic, are also going to be tolerant to ciprofloxacin. They're also tolerant to rifampicin. So this is going to be a whole different problem. If you were to take these persister cells and treat with vancomycin originally and get this population down to here, and then add another antibiotic, you might see a little bit of a drop-off, but you're still going to have some persisters survive. Resistance, on the other hand, if you were to add an antibiotic, kill off most of your population, and then add a different antibiotic, you would probably see eradication of that population. Because most of the time, the mechanism for antibiotic resistance doesn't provide resistance to multiple classes of antibiotics. And this is helping illustrate that idea of biofilms a little bit more clearly. So again, it's a community of bacteria adhering to each other in some kind of surface. So up here, we have a community of bacteria that were grown in the lab, and you can physically pick up that map of bacteria. You can see how robust this is to physically pick up that bacterial population. And then if you look at this on an individual cell basis, you can see your individual bacterial cells here, and then all this stringy material here. That's the biofilm matrix that's holding all of these cells together and also to some kind of surface. We also have a prosthetic device here. This should be completely clear, but you see all this yellowish and brownish color. This is actually a biofilm caused by a fungal species, Candida albicans. And you can see how this would be a problem if this were inside of you, right? That's a lot of bacteria. This valve, there should be a valve here. It's actually clogged from the fungus and the biofilm. And then a lot of times we see this in prosthetic devices. So you have some kind of prosthetic surface or some kind of foreign device like a catheter or a shunt. You have your bacteria growing on the surface of that, and then that's kind of wedged between the bone. And depending on the organism, they can secrete virulence factors that will actually degrade that bone and cause destruction of that bone tissue there. Now, on the right is a schematic of how we think persisters fit in with these biofilms. So this yellowish color is your biofilm, and then you have your individual cells within the biofilm. And in red are your persister cells, or those that are tolerant to the antibiotic. If you were to add an antibiotic to this, it would penetrate the biofilm, kill all the non-persister cells, and you're left with just a few remaining persister cells. Looks like the infection's gone away, so therapy is discontinued, and eventually a few months later, they can repopulate that biofilm and cause infection again. Clinically, what this looks like is they can't get rid of the infection in about 20% of patients, those relapsing patients. So eventually what they have to do is physically take out the prosthetic device, maybe the knee replacement or the hip replacement. The patient's on bed rest for 1 to 2 weeks while they're on antibiotic therapy, completely clearing the infection, and then they have to put in a new prosthetic device. Now, with this model, there's a couple things that aren't fully decided on in the literature. One, it's debated how well antibiotics can actually penetrate the biofilm. There's some literature suggesting that antibiotics can't penetrate the biofilm, although that's not the prevailing theory. And we actually worked with Dr. Lukey in the biology department, and he did confocal staining for us. And in this picture here, we have a biofilm that's caused a polymicrobial biofilm, so it's 2 species. It's that Candida albicans fungal species and then the Staph aureus bacterial species, compared to a biofilm that's just formed by Staph aureus alone. In green is vancomycin that's been fluorescently labeled. So we were doing this so we could see how well can vancomycin penetrate the biofilm. And you can see it's green all throughout the biofilm, maybe a little bit less green in the deeper layers of the biofilm, but vancomycin can penetrate the biofilm. So the reason biofilms are notoriously difficult to treat isn't because of antibiotic penetration, it's something else. On the bottom, we just used another antibiotic, delafloxacin. Its fluorescence is kind of weak, so it's difficult to see. It's in blue. But you can kind of see that blue color penetrating all the way down to the basal levels of the biofilm. The other thing that's not really known with this model is how does the immune system interact with these persister cells? So if you have an infection outside of a biofilm, after antibiotic treatment, you kind of rely on your immune system to clear the remaining persisters. That's been speculated. It hasn't really been shown how the immune system really interacts with these persister cells, or whether that biofilm protects those persister cells from the immune system. And I'll get into that in a little bit later in my talk. Outside of a biofilm, I mentioned previously that staph often causes what are known as minor skin and soft tissue infections. And this is actually from one of my friends who was getting her PhD at the same time I was, in the same lab that I was. And she was 27 at the time. She's now 31, and she's been getting these staph infections ever since, off and on. She can't get rid of them. And this particular one occurred over a month. it starts off as what looks like a minor pimple, right? Maybe a little bit more redness around it than you would think. Looks like something that should go away. And a lot of times she was getting these on her hands, her fingers, her thighs. This particular one's on her thigh. And over the course of the month, you see more redness being associated with it. And eventually, the redness and swelling got so bad, there was swelling in her knee, there was swelling in her ankle where it was difficult for her to put her shoe on, and she could hardly walk on that leg because it was so painful. This particular one lasted a month. Some of them only last a couple weeks, but it caused destruction to the muscle, so she has a noticeable divot in her leg now. Doctors have tried multiple classes of antibiotics. They had her put a cream all over her body to try and decolonize her of staph. They've had her stop shaving for months on end, different towels. They've tried a lot of different things. Nothing seems to get rid of these infections until they just resolve on their own. She'll be fine from the infection for a few months, and then they'll come back. So, in theory, it should be treated with antibiotics. They're testing it for what antibiotics is this organism susceptible to, and it's staph, and it should be susceptible to vancomycin. It's not clearing the infection. So we don't know it, but it's thought that persisters might also be a problem there. And we see that persisters aren't just a problem with Staph aureus. In every pathogenic bacterial species where they've been examined, they found persisters, and they're becoming increasingly associated with chronic infections. We see them in E. coli, Salmonella, Pseudomonas, Mycobacterium tuberculosis. We see them in fungal species. So there's becoming a little bit more of appreciation for these persister cells and antibiotic susceptible infections, but not to the point where we should be yet. Most of the emphasis is on antibiotic resistance. Now with that, I want to transition a little bit into some of my own data from our lab, and I just want to highlight a few different projects in the lab. I don't want to include a ton of data, but the first thing we wanted to do was identify a mechanism for how these persisters form in Staph aureus. And I'm not going to go through all of that data, but a lot of it was done in my postdoc and continued on here at UNK. But what we found is that as bacteria are growing, whether it's in an infection or in a flask in the lab, they're consuming some kind of sugar source. That's going to be their main source of energy. Oftentimes it's glucose, it's brought into the cell, broken down through glycolysis, and that feeds through the TCA cycle. Well, if you knock out the TCA cycle or you have less expression of those genes in the TCA cycle, you're going to have less energy. What we're finding is persister cells are just those cells with less energy, less TCA cycle activity. Related to energy is membrane potential. So if you have low membrane potential, you probably have low energy. So one of the first things we wanted to do after identifying the mechanism of persister formation was see, are persisters a problem in clinical isolates? So these are isolates taken from patients, sent to us, and then we test, is there a difference in killing? Can we identify some kind of mechanism in these clinical isolates that leads to persister formation? These particular isolates were sent to us from New York University, from Bo Shawson. That's why they all have BS in front of them. It doesn't stand for something else, if that's what you're thinking. But we measured membrane potential, and we have our lab strain here. So kind of a smaller bar on this graph means that these cells have high membrane potential, or fewer cells with low membrane potential. That means this one in red here has a lot of energy. If you take that lab strain and treat it with a chemical called CCCP, that's going to make the membrane potential go away. That's just a positive control. Now most of our cells have low membrane potential. Take a TCA cycle knockout, another control, and most of those cells have low membrane potential. We then start looking at our clinical isolates. And I've color coordinated this, so all of those clinical isolates that have low membrane potential are shown in blue here. All those with few cells that have low membrane potential are shown in red. So the ones shown in red, in theory, have a lot of energy available to the bacteria. Those shown in blue, less energy, and probably are more dormant-like. We then chose an antibiotic that all of these organisms should react similarly to. So they all have the same MIC, or the minimum inhibitory concentration. This is often a test done in a clinical setting to see whether an antibiotic will be effective on bacteria. They all have the same MIC to vancomycin. They should all have the same amount of killing to vancomycin. So here we have a kill curve. Again, it's set up like that first graph I showed, where you have the number of bacteria on the y-axis. The higher up, the more bacteria. And then you look at this over time. What we notice, this is actually a pretty strong correlation, right? All of the isolates that had very few cells with low membrane potential have a lot of killing. Those in blue that have maybe more dormant-like cells have less killing. In a clinical setting, all they do is test for an MIC. All of these should respond to vancomycin. Well, in vitro at least, you have some isolates that have zero killing after adding vancomycin. Now, they're not growing in the presence of vancomycin, so you know resistance didn't develop. If resistance had developed, they would have grown to over 10 to the 9th. So this is pretty nice evidence that we need to be considering antibiotic tolerance more, especially in a clinical setting. But the next step that needs to happen that really no one's looking at is, do these isolates that respond poorly to antibiotics, do they lead to worse patient prognosis compared to these isolates? And that's something that we're interested in looking into, and I've been talking to a couple of infectious disease doctors at UNC, but that's going to be the next activity, especially next experiment that we need to do, really to get people to start to notice antibiotic tolerance in a clinical setting. Another clinically relevant environment that we wanted to investigate is biofilms. I've shown lots of data with biofilms. Well, how do persisters behave within a biofilm? Here you can see a bar chart essentially, and again, the higher up on the bar chart, the more bacteria that survive. When we grow these biofilm assays, we use a 96 well plastic plate, and in the bottom of that, there's going to be wells where the biofilm forms. You add your antibiotics, let that go for 24 hours, then you determine how many bacteria were able to survive. Shown in blue is that TCA cycle knockout, which forms more persisters. So you essentially have a strain that forms a lot of persisters, and if you look at it in these various antibiotic treatments, you notice that it survives better when treated with ciprofloxacin, vancomycin, and then combinational treatments. So if you have a strain that forms more persisters, it does better in a biofilm. We then reasoned that biofilm cells, which are notoriously difficult to treat, may have lower energy than cells growing maybe in a flask. So we looked at membrane potential, and essentially with these graphs, if the population falls within this circle, it has low membrane potential. Shown in blue on each of these is our control that's been treated with that CCCP chemical. In red, on the first one, is cells growing in a flask. None of these are falling within that circle, which means these cells probably have a lot of energy. They're probably going to be pretty easy to treat with antibiotics. However, cells growing within a biofilm have low membrane potential, are going to be harder to treat. Another project in our lab is looking at polymicrobial biofilms. So biofilms are difficult to treat and have a high mortality associated with them. Well, now you add in another species, they have even higher mortality associated with them and longer hospital stays associated with them. And it's not really known why this is. There's been a lot of prevailing theories. One is it's increasing antibiotic resistance. Most of the field doesn't accept that as an explanation. Antibiotic diffusion, which has been being published. Again, we really didn't see that in things that antibiotics have poor penetration through the biofilm. And then there's been a few papers on antibiotic tolerance, but no one really identifying a mechanism for that. So the first thing we wanted to do is first just confirm that polymicrobial biofilms really are harder to treat compared to monoculture biofilms. So shown in red are polymicrobial biofilms, and these are again formed with Candida albicans and Staph aureus. And then you have Staph aureus alone biofilms shown in blue. And what you'll notice is with a lot of these antibiotics, ciprofloxacin, oxacillin, rifampicin, and then these combinational antibiotic treatments, you have more survival of Staph aureus when it's grown in the presence of Candida albicans. And this wasn't that surprising. Previous literature had shown that polymicrobial biofilms are more difficult to treat compared to biofilms caused by a single organism. However, we didn't really have a mechanism, and we wanted to apply some of what we were learning in our persister field to this polymicrobial biofilm field. And if you remember me mentioning that glucose is a common source of energy for bacteria to take into the cell, break down, and have energy to grow on, well, we looked at glucose consumption between staph growing by itself, which is shown in blue, and then staph growing in the presence of Candida albicans. And you'll notice that staph growing in the presence of Candida albicans is consuming glucose more quickly. So what this means is, yes, the fungal species is probably consuming some of that glucose. It's using the nutrients from the environment more quickly. And then in later phases of growth, when they start secreting virulence factors and actually causing damage, they're going to be pretty low energy and more dormant-like and probably harder to treat. And we just confirmed that by actually measuring the amount of energy in those cells. Staph grown in the polymicrobial biofilm had less energy than staph grown alone. So this is offering a specific mechanism that might help explain why these polymicrobial biofilms are so difficult to treat. Shifting gears a little bit, if you remember from that model early on, that biofilm model, where I said there's a couple things that we didn't really know about the model, and one of those was how persister cells are interacting with the immune system. Well, our body has what are known as antimicrobial peptides. And these are kind of, to put it in simplified terms, our body's own natural antibiotic. They resemble antibiotics in some ways. They're short cationic peptides, and a lot of times they kill bacteria either by forming pores or having similar targets as antibiotics do. So they can specifically target replication, transcription, translation, maybe cell wall synthesis. For these reasons, we thought, well, it would make sense if persisters are not only a problem with antibiotics, but maybe they're also a problem for our innate immune system. Also, it's been shown that pore formation is, or theorized anyways, there's not a lot of evidence for it, but it's been theorized that pore formation that antimicrobial peptides use to kill bacteria is partially dependent on membrane potential. So if we have persister cells that have less membrane potential, and a lot of targets of antimicrobial peptides are similar to antibiotics, it would make sense that persisters can survive these antimicrobial peptides better. And so Alexis, who's sitting right here, did some of these, nice shout out, did these assays where she took 2 known antimicrobial peptides that target Staph aureus and took a TCA cycle knockout and looked, if you have a TCA cycle knockout that forms more persisters, does it survive better to these antimicrobial peptides? And that's shown in blue. And yes, you have more survival compared to your wild type Staph aureus strain shown in red. Same with human beta defensin 3. And then our control over here, there's really no difference between the 2 strains. So we're seeing these persisters are not only a problem for antibiotics, but also antimicrobial peptides. What about other parts of our immune system? And so we wanted to look at starting off with macrophages. So after you get an infection, you're going to have recruitment of immune cells, neutrophils, macrophages. They're going to engulf the bacteria, kill the bacteria. We wanted to know, can these persister cells survive better within a macrophage compared to wild type? And both of these are pretty preliminary, but we're excited by this data because what we see is in a TCA cycle knockout, you have better survival compared to your wild type Staph aureus. And ways that macrophages can kill bacteria, it could be through producing reactive oxygen species, reactive nitrogen species, maybe a low pH. We tested all of those, and really the one that's shown the most promise for why persisters are surviving better within a macrophage is reactive oxygen species. So if you take a chemical called paraquat, this is going to induce reactive oxygen species. Here, our TCA cycle knockout is surviving that paraquat treatment better than our wild type strain. So it seems that these persisters are not just a problem with antibiotics, but they may also be a problem for our immune system. And we're preliminary in this process, but we're encouraged by this data. To summarize, and I'll just go through this pretty quickly, but shown that, and I didn't have time to show it, but persisters are formed through low TCA cycle activity. We've seen that in clinical isolates, Low membrane potential correlates to a high persistor phenotype or the ability of antibiotics to not work as effectively as those with high membrane potential. We see persisters being a problem within biofilms and polymicrobial biofilms, and then also they may present a problem to our innate immune system. And with that, I'd like to thank everyone who's helped out on this project. I mentioned Alexis already. Dan did some of that polymicrobial work. And then I have a collaborator at Northeastern. She's going to be finishing up her PhD this spring, Eliza. And then everybody else who's helped out on this project and funding. And then with that, so kind of an inside joke is we had a student presenting at a conference, and it was her first conference ever. She was a freshman, and we had a faculty member from another university come up to her and start berating her, saying that persisters aren't real, all of her data is meaningless. It kind of went on for quite a while, so it's kind of become an inside joke how persisters aren't real, all of our data is meaningless. Kind of fitting with that, germs aren't real, so we don't need to wash our hands. Thank you.
So do we have any questions for us?
I'm about as far away from a biologist as a chemist can get. But so you've got a way that you can treat a cell that turns high membrane or a high potential membrane into a low potential. Can you do the opposite?
Treat and make a low membrane potential cell a high membrane potential cell. There's been a little, some research trying to kind of waken up these persisters. They haven't specifically looked at membrane potential, but what they've done is supplement carbon sources like glucose with the antibiotic, so giving them more nutrients essentially, hoping those bacteria will sense those new nutrients there and start growing again. It's had limited success. It's had more success in E. coli than staph. They didn't specifically look at membrane potential, but maybe something along those lines to just try and make the cell more active essentially.
I'll ask you my question, not Darby's question. You said that when the persisters have a reaction to the antibiotics, it's kind of a non-specific kind of reaction. Does it matter if they're gram-positive or gram-negative?
No. Persisters by nature are dormant-like, and so they don't have active targets. So for most antibiotics to work, they have to incorporate into an active target. So the cell wall, you need to be synthesizing cell wall for the antibiotic to actually bind and incorporate, or making proteins. And persisters by nature are more dormant-like. They're not really growing or as quickly, and so there's really not a place for the antibiotic to incorporate into. There are some antibiotics that are energy independent and can form pores into the membrane, like daptomycin. But we tested daptomycin and actually found that persisters are more tolerant to daptomycin as well. And what we found is the reason is daptomycin pore formation depends on membrane potential. So if you briefly treat the cells with that CCCP before and make the membrane potential go away, daptomycin is no longer effective.
All right, well, if there are no other questions, again, thank you everyone for coming out, braving the cold and the snow. Again, let's thank Austin for this great presentation.